Hello everyone. So hello everyone. Good morning everyone. So today we are going to discuss about the Neet question paper. Okay. So this is a test which is conducted on 19th June. So today we are going to discuss about the biology. Okay. So today we are going to discuss about some of the important questions. Okay. Fine. So let us start the questions. The question number one is, which one of the following structure is developed into the seed coat after the fertilization? Okay, so they are telling that which structure will form the seed coat after the fertilization. Okay, so here they are given the option one is hilum, and option two is panicle, and option three is the integument, and option four is micropyle. So before going to answer this question, so let us see the seed structure. So if you see the seed structure, so you can see the seed coat here, right? So the outer you can see the seed coat, and inner side you can see the stored food. So it will also call it as an endosperm, right? So the endosperm is helps in the growth of the, it will give the nourishment to the embryo, growing embryo, right? So here you can see the growing embryo, right? Fine. So let us see about the options which are correct. Okay, fine. So let us go to the first option. They are telling that hilum. Okay, so the hilum, so hilum is not the correct answer because so here hilum, so it connects the, okay, so hilum helps in the connection of, it connects the ovule to the ovary, ovule to the ovary. Okay, so here you can see the hilum. So it is helps in the connection of this ovule to the ovary. Okay, so that is the reason option A, option one is wrong. Okay, so then coming to the option two, so that is a funicle. Okay, so option two is a funicle. So here you can see the funicle. Okay, funicle is a filamentous like neck-like structure. Okay, so it is a neck-like structure. It helps in the, so connection between, it helps in the connection between ovule to the placenta, ovule to placenta. Okay, so it helps in the connection between the ovule to the placenta. Okay, fine. So let us go to the fourth question. So that's why the option two is also wrong. So then coming to the fourth option, so they're telling that micropyle. Have you observed? So during the fertilization, so when the fertilization is happening, so on that time, so the pollen tube will be formed, right? Either it can form from the chalazal end or it can form from the micropylar end. So most probably it will form the micropylar end, right? So here you can see the micropylar end. So where the pollen tube will be formed from that. Okay. So but it's not the correct answer. Okay. So that is why option four is also not the correct answer, where pollen tube will enter from here. Okay. So it is helps in the pollen tube entering. Pollen tube entering. Okay. So that is the reason option four is also wrong. The correct answer is option three, right? Yes. So the correct option is option three. So that is the integument. Okay. So the integument will form the seed coat. Integument is the outer coat. It will form the seed coat. Okay. After the fertilization. Here you can see. So this is the integument. So that is the outer coat. So that will form the seed coat. Okay. It is a thick in structure. Fine. So let us go to the next question. This question is from the chapter. So there is a population and interaction. Okay. The question is decomposition of detritus. Okay. All of you know that what is the decomposition, right? Decomposition is nothing but, so breakdown of larger molecule into the breakdown, right? Breakdown of large molecule into the small molecule. So the large molecule into the smaller molecule, right? Smaller molecule, right? So and there here as they're asking about the detritus. What do you mean by detritus? You can take any of the fallen leaves, so that will be the detritus, or any dead organism. So death of the any animal, so that also you can take the detritus. Okay. So here they're asking that. So which one is the odd one? Okay. So we have to remove the odd one. Okay. And sorry. So they are telling that. So which one is the correct one related to the decomposition? So here they are giving the so many odd things. So odd statements are given. In that you have to choose the correct option. Correct option. Okay. So let us see first option. It is a fast when detritus was rich in lignin and chitin. No. So whenever the whenever any substance that is made up of the lignin and chitin, it is a polysaccharide, right? So it is a polysaccharide. So obviously the decomposition will be slow it will be, okay? So it is a polysaccharide. So the decomposition will be slow on that time, okay? So that is the reason it is not fast, it is a slow, okay? It will be slow, okay? So that is why option 1 is wrong, okay? So then coming to the option 2, it is a largely an aerobic process. Yes. So here the breakdown of the organic molecule is going to be taken place. Yes, it is a aerobic process, okay? So that is the correct. So then coming to the option 3, is slower when detritus was rich in the water soluble sugar substances. Water soluble sugar substances are the monosaccharide, right? So these are the simpler molecule, right? Simpler molecule. So when the detritus was rich in the simpler molecule, obviously the decomposition will be fast, okay? So it will not be slow, okay? So it is slow. No, it is a faster, okay? So it will be a faster. Remember, student, okay? Fine. So then coming to the, so that is the reason. So this option is also wrong. So then coming to the 4, gets faster when temperature is low. You all know about all know about the reactions, right? Whenever the temperature is high, the reaction will be fast, right? So here they are telling that it gets faster when temperature is low. No. So when the temperature is low, it will be slower, right? It will be slow, right? So when the temperature is more on that time, so the decomposition will also be more, okay? So that is the reason option 4 is also wrong. The correct answer is it is a largely an aerobic process, okay? Fine. So the option 2 is the correct answer. So let us go to the next question. Which one of the following bonds linkages are not present in the DNA? Okay, so you all know about what is the DNA, right? So DNA is nothing but the deoxyribonucleotide, right? Ribonucleic acid. Deoxyribonucleic acid, right? So which are the bonds present in the DNA? So before going to talk about DNA, first let us see what are the things which are present in the DNA, okay? So which are the things, guys? So the first one we can see the nucleotide. So the nucleotide is made up of the pentose sugar, right? Pentose sugar, and it is made up of the nitrogen bases, right? Nitrogen bases, and third one is the phosphate group, right? Third one is the phosphate group. Okay. So this, including these three things, we will call it as a nucleotide, right? Including these three, we will call it as a nucleotide. So let us see the bonding. So the diagram here you can see, if I take this as a nucleotide, so this is a one nucleotide, this is the second nucleotide. Okay. So here you can see between these two nucleotides, the bond is present. Okay. So which is that bond? So this bond is there, right? So that bond, I will call it as phosphodiester bond. Phosphodiester bond. Phosphodiester bond. Where it is present? Between two nucleotide. Between two nucleotide. Right? Okay. So, and so that's why option 3 is a correct thing. It is present in the DNA, right? So here they're asking that which one is not present, right? So obviously phosphodiester bond is present. So that's why I'm eliminating that option. So then coming to the, have you know about, guys, the Chargaff rule? So A is equal to T, right? And A bond with the T and the G will bond, triple bond with the C, right? C. So G is triple bond with the C, right? Which is the bond present between these two? Hydrogen bond, right? So A is connected to the thymine, adenine is connected to the thymine by the double hydrogen bond, whereas the guanine was connected to the cytosine by the triple hydrogen bond, right? So that is why hydrogen bond is also there. Okay, so that's why I'm eliminating that also. Then coming to the N-glycosidic bond. Okay, so here you can see, so this is the pentose sugar, okay? This is the pentose sugar, and here you can see it was attached to the nitrogen base. Okay, so it is attached to the nitrogen base. So this bond is there, right? Where the sugar molecule was attached to the nitrogen base. So that we will call it as N-glycosidic bond. Okay. Yes, N-glycosidic bond is also there. So that is between the sugar molecule and the nitrogen base. Sugar molecule and the nitrogen base, right? So that is why, so this third option is also correct, right? So then they are asking about the peptide bond. No, we can't see the peptide bond in the nucleic acid, right? Because peptide bonds is present in a protein, right? It is present in the protein. It is not going to be present in the DNA, right? It is going to be present in protein. Okay, it is going to be present in the protein. So between the amino acid, right guys? So that is why the option 1 is the correct answer, because here they were asked about that. So which is not present in the DNA, right? So that is why. So I am removing the other options. Okay, so I think you understood this. Let us go to the next question. Select the odd one with respect to the detritus food chain. Okay, so you all know that there are the two types of the food chains are present, right? One is the grazing food chain and another one is the detritus food chain, right? So the detritus food chain will be more in the aquatic environment, so whereas the grazing food chain will be more in the terrestrial habitat. Okay, so let us learn about. So they are asking that. So you have to remove the odd one. Odd one means what, guys? So three statements are correct. One statement you have to remove. Okay, which one is not related to that? So whenever you are observing this type of the question, remember guys, so here this all the statement is related to the detritus food chain only, but remember the steps. Okay, so here you have to remove which is the not steps that related to this. Okay, fine. So let us start the answering. So you know that the detritus food chain will be start with the dead organic matter, it from the leaf or it's from the dead animal, right? Okay, so it begins with the dead organic matter. Yes, option 1 is correct, right? It start with the dead organic matter. So after that, what will happen? It is made up of the decomposers such as fungi and bacteria, right? Yes. So it is made up of the decomposers. So those fungi and bacteria will help in the breakdown of that molecule, breakdown of the large molecule into the smaller molecule, right? So that is also correct. So then coming to the fourth option, they are telling that saprophytes, you know, right guys, what is the saprophytes? Saprophytes are nothing but heterophytes, right? Heterophytes, they are dependent on the other molecule for the food, right? So then where is the difference came? So here they will secrete the enzymes outside the body, okay? It will secrete the, for example, saprophyte if you take means, so this is the organism, it will secrete the enzyme outside, okay? So it will secrete the enzyme outside, and here there is the food is there, okay? So here the food is there. So it will help in the, these enzymes will make the breakdown of this molecule. Okay. Breakdown of this molecule. So then this breakdown molecule will be smaller in size. Now it will be absorbed to this saprophyte. Okay. Yes. So what it will do, it will secrete the digestive enzyme to break down the dead material, and it will convert that into the smaller molecule, and it will be absorbed into the saprophytes. Okay. So that is also there. So that is also the correct answer. Okay, these all are the steps. Okay, so then coming to the third option, they are telling major conduit for energy flow among the aquatic ecosystem. So that statement is correct, but compared to these three statement, it is not suitable, right? So here they are given the step how the organic matter is going to be undergoing the step by step breakdown in the detritus food chain. But so here it is telling about the energy, right? So that is the reason option 3 is wrong, guys. Okay, option 3 is a wrong. So that's why here they are telling that odd one out. So that's why option 3 is a correct one. Okay, 1 minute. Yeah. So option 3 is a correct one. So the others are wrong. Okay. Okay, fine. So let us go to the next question. All of the given are correct for the logistic growth of the population except, okay, so here also you have to remove the odd one out. Okay guys, it is very easy to remove the odd one, right? Yes. So see here, so in the option 1 they are telling that shown by the population. Guys, this chapter also belongs to the population and the interaction. Okay, so all the given are correct for the logistic growth. Okay, So the shown by a population growth in a habitat with a limited resources. Okay. So you know about the logistic growth, right? So when you're talking about the logistic growth, so the logistic growth is the realistic, right? It is the realistic, right? Realistic. And coming to the logistic growth, so it will be happen when the it is having the limited source. Okay, it is having the limited resources. Limited resources, right? Okay, let us see the question here. So, shown by the population growing in a habitat with the limited resources. Yes, so that is correct. So, have you remember? So, if it is unlimited resources means, so that is come to the exponential growth, right? Where the resources unlimited there, right? In the exponential growth. But in the logistic growth, it is limited. So that's why that is the correct option. So then coming to the option 2, lag phase is followed by log phase and then deceleration phase. So remember guys, deceleration phase is nothing but stationary phase. Okay. It is nothing but stationary phase. Stationary phase. Let us learn about the graph. Okay. So how we will do the graph here? See here, if it is a logistic growth here, so we'll write like this, right? So here I will write the time on the x-axis. On the y-axis, I will write, so there's a population, right? I will write the population number, right? Population number, right? So how the graph will be grow, student? So it will grow like this, right? Right? So it will grow like this, right? So yes. So observe here, what are the things here? Okay. So starting the growth will be very slow. Okay. So it will start with the slower process. Okay. So that's why we will call it as this is the lag phase. Okay. 1 minute. One minute. So it will be like this. Okay. Yeah. So the starting, what will happen? So it will start with the very slow process. Okay. So that's why we will call it as a lag phase. Remember, lagging. Lagging means what? It is a slow, right? If any person is walking, he was walking very slow, means what we will call it as? Why you are lagging, right? So lagging behind, right? Yes. So the lag phase will be slow. Okay. It will be slow. So then later, so the growth will be very fast. Okay. The growth will be multiply very fastly. So that phase we will call it as a log phase. Log, L-O-G, log phase. Okay. So then what will happen? The growth is happening. So sometime when the growth was happened, so multiplication was happening very fastly. On the time what happened? So the sources become limited, right? The sources become limited on that time. So on that time what will happen? The growth is stopped, right? The growth will be stopped on that time. So that is why it is reaches the stationary phase. We will call it as stationary phase. Stationary phase, right guys? So that is the reason option 2 is the correct answer, right? Yes. So option 2 is the correct. So then coming to the option 4. Option 4, it is telling that it is considered to be the more realistic one. Yes, it is a more realistic one, right? I told you that before only, right? So when you're talking about the exponential growth, so that is a theoretical one. So coming to the logistic growth, it is a realistic one, right? So that is also correct. Fourth one. So then coming to the option 3, they're telling that so it is a J-shaped curve. No, it is not a J-shaped curve, right? It is a S-shaped curve, right, guys? So it is a S-shaped. Here only you can see the shape of the curve is S-shape, right? The shape of the curve is S-shaped, and we also call it as a sigmoidal curve. Sigmoidal curve. Sigmoidal curve, right? So that is the reason. So here they are telling that which one is the correct for the population growth. The correct one is option 1. 1 minute. So which one is the correct for the logistic growth? Which is the except, right? Except they are telling which is the odd one out. Odd one, right? Yes. So option 3 is the correct answer, right? So because what happened? So here it is not it is the S-shaped curve, right? So option 1 is correct. option 2 is correct and option 4 is also correct. Okay, guys. So the correct answer is option 3, right? All 1, 2, and the 4 statements are right. Okay. So let us go to the next question. The question is read the statement. Okay. So read the statement and select the correct option. Okay. So you have to read the statement A and statement 2 and select the correct option, which is in the 1, 2, 3. Life cycle of the parasites are often common. So do we have only one or two intermediate hosts? Yes. So if in the two hosts, right? One is a human and another to facilitate the parasitation. We are talking about the Plasmodium vivax. So which one is the primary host? Primary host means we will tell that is a mosquito. And so on which host it dependent on the nutrition? It is not dependent on the nutrition for the mosquito, right? On which it is dependent on? It is dependent on the humans, right? So that is the reason. So human we will call it as the clinical host. Okay. So where the parasiting. So here the vector is what? Mosquito, right? Mosquito, it is acting as a vector for the parasitization of the plasmodium to the host, right? So that is the correct reason. Okay, option A is the correct only. Then coming to the option 2, statement B, they are telling that majority of the parasite harm the host, right? And they may reduce the survival, growth, reproduction of the host and reduce the population density. Yes. So whenever any person was attacked by the any type of the parasite, first it will reduce the growth of that person. Okay, it will reduce the growth. Okay. So the later reproduction is going to be inhibited, right? So then later the survival, right? If any person was attacked by the any parasite, he is going to be live for the 100 years? No, right? Because the nutrition was taken up by this organism only, right? So that is the reason, guys. So the option B is also wrong. Sorry, option B is also correct, right? So the host and the reduce its population density. So it is goes on reducing the population density of the host, right? So that is the reason option 2 is also correct. Okay. So that's why option 1 and option, sorry, in the option 1, they are telling both A and B are correct. So that is the correct reason. Okay. Yes. So A and B is correct. Okay. Then coming to the next question. Okay. Yes. So that is the reason option 1 is the correct answer, whereas the option 2, option 3, and option 4 are the wrong option. Okay. Fine. So let us go to the next question. Which one of the following feature is not related to the facilitated diffusion? Okay. So you all know that. So what is the facilitated diffusion, right? So this will comes under the chapter that is transport in the plant and also in the chapter cell. Okay. Fine. So you all know about diffusion, right? For the diffusion, we don't require any type of the protein molecule, right? Yes. So, but what here they're telling facilitated diffusion. So the diffusion is facilitated by some molecule, right? So which is that some molecule? Nothing but our protein only, right? Yes. So let us talk about the statements which are given here. In the statement 1, they are telling that transport proteins are required in it. Yes. So the transport proteins are required in it, right? Yes. So you can see here. So this is a plasma membrane. If you take this is a plasma membrane mean. So in the plasma membrane, we can see the integral protein, right? So have you seen, guys, there are the two types of the proteins are there. One is the integral protein and another one is the peripheral protein, right? So here, this integral protein is helping in the transport of the water soluble molecule. If it is a lipid soluble means it can easily pass through the plasma membrane, right? But so here there only the water soluble molecule will be transported by the protein molecule. Okay. Remember, guys. So the molecule should be water soluble. Okay. So the molecule should be water soluble. Then only it can be transported. Okay. Water soluble. If it is a lipid soluble means it can easily diffuse through the plasma membrane. Right? Yes. So that is the reason it is a water soluble and transport proteins are required in it. Yes, correct. The integral proteins are required in it. Integral proteins are required, right? Okay. So in the option 2, it occurs along the concentration gradient. Yes, guys. So whenever we are talking about the diffusion, so diffusion will be like along the concentration gradient. See here. So here the molecule is a more in concentration, and here the molecule is very less in concentration. Okay. So here the transport will be happen. Okay. So here the transport will be happen from the higher concentration to the lower concentration, right? So if it is from the higher concentration to the lower concentration, from the high to low concentration means we will call it as along concentration gradient. Along concentration gradient, right? Along concentration gradient. Okay. So, but here what will happen? So the along the concentration gradient only it will also happen. So that is the reason it is correct. Okay. So then in the option 4, they are telling, in the option 3, they are telling that it requires the energy in the form of ATP. No, guys. So for the facilitated diffusion, the name is telling only diffusion, which does not require any type of the ATP molecule. Okay. That is a wrong. So then where the ATP molecule is required? In the active process, right? In the active process, we require the ATP, right? So where the active process is nothing but against the concentration gradient, right? Against concentration gradient. Remember, guys. So whenever the active process is happening, so their ATP molecules is also required and the protein molecule is also required. But so here the molecule will move from the lower concentration to the higher concentration. So that is why we will call it as against concentration gradient. So how the against concentration gradient will take place? For example, this is a plasma membrane. So there is a protein molecule. So here the molecule's concentration is very less. Okay. So here the molecule concentration is very high. From here to from the lower concentration to the higher concentration, the molecule is moving. That we will call it as a against the concentration gradient. Okay. So that we can see in the active process, right? So that is a not correct answer for the facilitated diffusion. Okay. Then in the fourth option, they are telling it shows the response to the protein inhibitor. Yes, guys. So whenever the protein is there, so if any inhibitor bind to this, okay, any inhibitor bind to this means it is not able to transport the molecule from the higher concentration to the lower concentration, where protein is going to be inhibited, right? So that is the reason option 4 is also the correct answer. Okay. So then which one is the wrong answer here? That is a wrong statement. It requires the energy in the form of ATP. So that is a wrong, right? So here also they are telling that is not related to the facilitated diffusion, right? So that is the reason option 3 is the correct answer. Option 3 is the correct answer, whereas the other statements are correct, but in the option those are wrong. Okay. So fine. So let us go to the next question. Eighth question. So which one of the following is not true for the glycolysis? Okay. So let us talk about the glycolysis. All of you know that glycolysis is nothing but where the one glucose molecule was broken down into the 2 pyruvate molecule, right? It is break down into the 2 pyruvate molecule, right? So let us go to the option statement number 1. Hexokinase convert glucose to the glucose 6 phosphate. Yes. So this is the first step. Yes, it will convert the glucose into the glucose 6 phosphate. That is correct. Then coming to the NAD plus is reduced to the NADH plus H plus. Yes. So it will also take place, right? So then coming to the option 3, through the substrate level phosphorylation, net gain of ATP is 6. So first we had to understood that in the substrate level phosphorylation will be happen in the step number. Okay. The substrate level phosphorylation. Substrate level phosphorylation. So what do you mean by this substrate level phosphorylation means? The phosphate which is present in the substrate that is utilized for the synthesis of the ATP. Okay. So substrate level phosphorylation. In the glycolysis, there will be 2 substrate level phosphorylation will be happen. So one is at the seventh step and another is at the 10th step. Okay. In the 7th step, you can see that 1,3 bisphosphoglycerate. Bisphosphoglycerate is converted into the 3-phosphoglycerate, right? It is converted into the 3-phosphoglycerate. So what will happen there? So ADP for the substrate, ADP and PI. So there, the phosphate group is here, right? In the first position. So that will be utilized and it converted into the ATP. Okay. So that is the 1 ATP. And in the 10th step, what will happen? So there is phosphoenol pyruvate is there, right? Phospho, 1 minute. Phosphoenol, okay. Phosphoenol pyruvate is there. Phosphoenol pyruvate, it is converted into the pyruvate, right? It is converted into the pyruvate by, here you can see ADP plus PA is there. By utilizing the phosphate group in the substrate, it is converted into the ATP, right? So there are 2 ATP molecule is produced, right? So that is the reason option 3 is also wrong. It is not a 6, okay? It is a 2 ATP, right? So that is the reason option 3 is wrong. So then coming to the option 4, the 2 pyruvic acid molecules are produced from the each glucose molecule. Yes, from the each glucose molecule, I have told you that 2 pyruvate molecule is going to be produced, right? So that is the correct statement, right? So here they're telling that not true. Here I have to remove the odd one, right? So that is the reason option 3 is not the related to the glycolysis, right? So that is why option 3 is the correct answer. Option 3 is not related to this, and whereas the other statements are correct for the glycolysis. Okay, fine. So this is question is correct. So then coming to the next question. So this is a ninth question. During the electron transport chain in the mitochondria, so what will happen? Okay. Option 1, they are telling, so whenever you observe the question, so see here what they are telling that during the electron transport chain in the mitochondria. So here you have to select the one correct statement. Okay. So here they are given 3 wrong statement. So here you have to select the one correct statement. One correct statement. Okay. So let us search for the which is the one correct statement. Okay. So electrons are donated by H2O. No. So in the photosynthesis, it is donated by water. But in the mitochondria, it is donated by NADPH, right? Sorry, NADH and FADH2, FADH2, right? It is not by the water. So that is the reason option 1 is wrong, right? So then coming to the option, sorry. So let us cut here, 1 minute. Yeah. So that is wrong, right? So then coming to the option 2, they're telling that the terminal electron acceptor is the oxygen. Yes. So the terminal electron acceptor is the oxygen. So then it will convert it into the water, right? So oxygen later it will convert into the water, right? Yes. So that is also correct answer. So that is the correct answer, option 2, right? So option 2 is the correct answer, right? So then coming to the third statement, NADH dehydrogenase accept the electron from ubiquinone. No, the name only telling that NADH dehydrogenase, it will accept the electron from the NADH, right? NADH, not from the ubiquinone, right? So it is the first electron acceptor or the complex number 1. So it will accept the electron from NADH, not from the ubiquinone. So that is the reason option 3 is wrong. So then coming to the statement number 4, the electrons are transferred from the complex 1 to the complex 2. Someone will think that yes, correct, but not correct, right? So if you take the mitochondrial membrane, inner membrane, mitochondrial inner membrane. So if you see, so this is the complex number 1. Okay. So this is the complex number 2. So this is the complex number 3. And this is the complex number 4. Okay. This is the 4. And this is the 3. And this is the 2. And this is the 1. Okay. So where complex number 1, we will call it as the NADH dehydrogenase. So it will take the electron from the NADH and it will send the electron to the complex number 3, not the complex number 2. Okay. So it will send to the complex number 3. Okay. So obviously the complex number 2 will accept the electron from the FADH2. FADH2, right? We will also call it as a succinate dehydrogenase, right? Succinate dehydrogenase, complex number 2, right? Dehydrogenase, right? So it will accept the electron from the FADH2. Then it will also send the electrons to the complex number 3, right? So that is the reason. So then from the complex number 3, the electrons will be transported to the complex number 4, right? So that is the reason the complex number 1 will not transport the electrons to the complex number 2. So that is the wrong option, right? So the first bill electron will transport it to the ubiquinone, right? So there is a ubiquinone is there, ubiquinone. From the ubiquinone, it will transport it to the complex number 3. Okay, from the ubiquinone, it is transported to the complex number 3. So that is the reason. So here also, FADH also first it will send the electron to the ubiquinone. So then it will send to the complex number 3. Okay, fine. So that is the reason this option is wrong. Okay, so it will send to the first ubiquinone, then it will send to the complex number 3. Okay, fine. So that is the reason option 1, 3, and 4 are wrong. So the correct option is option number 2. Okay, fine. So let us go to the question number 10. Here they're asking that, so number of the carbon dioxide molecule used for production of sugar polymer. The sugar polymer means what, guys? Polymer means, see here, one glucose molecule, second, third, fourth, fifth, sixth, seventh, eighth. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so this is a polymer of the sugar molecule. So it is a polymer of glucose. So here each one is a glucose here. Okay, each one is glucose molecule here. They're asking that. So this is formed by the Calvin cycle. Okay, Calvin cycle is nothing but dark reaction, right? Dark reaction. Where this dark reaction is going to be taken place? So it is in the stroma, right? In the stroma. So now you tell me, so how many carbon dioxide molecule required for the 10 glucose molecule to produce? First, let us think about for the one glucose molecule. Okay, 1 minute. For production of one glucose molecule. Okay. So first let us talk about the molecular formula of glucose, that is C6H12O6, right? For formation of the one glucose molecule, I require a 6 carbon dioxide, right? So then obviously, so 10 glucose molecule means 10 glucose molecule means how many carbon dioxide I required. So we can do the cross multiplication. 6 into 10 divided by 1 is equal to 60, right? Yes, everyone you can give the answer. So that is the option 3, right? Yes. So option C is the correct answer, where option 1 and 2 and 4 are the wrong statement. Okay, guys. So the 60 molecules will be utilized for production of the 10 molecules of the glucose. Okay. Okay. So then coming to the next question. So this is from the human physiology. The question is, which one of the following statement is correct in relation to the endocrine system? Okay. Here they're asking that. So correct statement. Okay. So what do you mean by correct statement here? So they are given here. So 3 wrong statement. Okay. 3 wrong statement they are given. Here you have to correct. Here you have to select the one correct statement. Okay. Fine. So let us go to the first question. Organ in the body like gastrointestinal tract, nothing but the GI tract, and heart and kidney, liver will not produce. Remember here, do not produce any hormone. Is that correct, guys? The statement is correct? No, right? Because our heart, kidney, and liver produce the hormones, right? Our heart will produce the ANF hormone, right? Natriuretic factor. Then coming to the kidney, it will also produce the hemopiotic hormone, right? So that is also helpful in the production of the red blood cells, right? So that is also produce the hormone. Then liver also produce the hormones, right? But here they are telling that do not produce any hormone. No, it will produce the hormone, right? So that is why this statement is wrong. Then coming to the option 2, hormone are non-nutritive chemical. They will not a non-nutritive like the protein, okay? Produced by the body in the trace amount. So that act as the intracellular messenger. So it's termed as a hormone, right? So that is a correct statement, right? So it is a non-nutritive substance that is act as a intracellular chemical messenger. So that is the correct statement. So then coming to the option 3, releasing and the inhibiting hormone are produced by the pituitary gland. No, releasing and the inhibiting hormones are produced by hypothalamus, right? So those are produced by hypothalamus. Hypothalamus, right? Releasing and inhibiting hormones are produced by hypothalamus, not the pituitary hormone, not the pituitary gland, right? So that is the reason this option 3 is also wrong statement. Then coming to the option 4, so adenohypophysis is under the direct neuronal regulation of the hypothalamus. No. So whenever we are talking about the hypothalamus, so we have the pituitary gland, right? Pituitary gland. So pituitary is having the two parts, right? So which are those two parts? One we will call it as the anterior, right? So anterior part. So that is under the chemical condition, right? Chemical coordination, that is having under the chemical coordination. So that we will call it as an adenohypophysis. 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 It is not the direct control. Okay. So then coming to the posterior one, posterior. Okay. Posterior we will call it as a neurohypophysis. 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 Have you remember, guys? Oxytocin and ADH is produced by the posterior part, right? Yes. So it is not actually produced by the posterior part, but it is produced in the hypothalamus, right? Hypothalamus, then it is released by the posterior part, right? So that is the direct control of the neuronal, right? Neuronal of the hypothalamus. So that is the reason. So neuronal regulation of the hypothalamus. So that is the reason option 4 is also wrong, right? So not the adenophysis is under the neuronal control. The neurohypophysis is under the neuronal control, right? Remember, guys. So that is the reason option 4 is wrong. Okay. So option 4 is wrong and option 3 is also wrong. So then coming to the option 1 is also wrong. So the correct statement is option 2, right? Okay. So option 2 is the correct answer. So then coming to the next question. Sulfonamide drugs are used in control of the bacterial pathogen is an example of. So this will come in the chapter of biomolecules. And remember, guys, so here the sulfonamide drug. Okay. So the sulfonamide drug. So have you learned about the enzymes, guys? In the enzymes, you can talk about the inhibitors, right? So enzyme activity is going to be inhibited by the inhibitor, right? So here they are telling that non-competitive inhibition. No, it is not a non-competitive inhibition. Non-competitive inhibition will what happen means here the inhibitor will not inhibitor will not resemble the substrate. Resemble substrate. Okay. So that is the reason not the non-competitive inhibitor. And coming to the feedback inhibition. So where the final product will inhibit the enzyme. So that we will call it as the feedback inhibition. So final product will inhibit the enzyme. Okay. So that is the reason option 3 is also wrong. So then coming to the allosteric modulation. So do you know what is allosteric modulation, guys? So here, for example, ATP. Okay. So it will bind other than in the allosteric modulation, what will happen? Which is the inhibiting molecule, it will bind other than active site. But it will modulate the active site of the enzyme so that the substrate will cannot bind to that active site. So thereby it is going to be inhibit the enzyme activity. Okay. So that is the reason option 4 is also not the correct answer. Other than active site, right? Okay. So then coming to the option 2 is the correct answer. So that is the competitive inhibition. Okay. So what happened in the competitive inhibition is, so it will resembles the PABA. Okay. It resembles PABA, PABA. Okay, PABA means p-amino butyric acid. P-amino-butyric acid. Okay. So there it will resembles the PABA by competitively it inhibits the enzyme. So which is involved in the folic acid synthesis. Folic acid synthesis. folic acid synthesis. Okay, so that is the reason. So the correct answer is option 2. Okay, sulfonamide drug will be competitive inhibitor. Okay, so it will inhibit the folic acid synthesis in the bacteria, and we will also call it as sulfonamide is a broad spectrum antibiotic. Broad spectrum antibiotics. Antibiotics. Okay, guys. So I think you understood this question. Let us go to the next question. The event that finally leads to the formation of the crossbreeds in the muscle contraction. So just recall the chapter. So that is the locomotion and the movement. Okay. In the chapter, so the release of the calcium from the, which is the first thing will happen from the motor neuron. The from the motor neuron, it will carry the neurotransmitter that will release it, right? When it is released, so it will release the calcium ion. So then from the sarcoplasmic reticulum, that is the first step, right? So when the neurotransmitter is released, so there the excitation will happen. So when the excitation will happen, so the calcium ion going to be released from the sarcoplasmic reticulum, right? So that is the first step. That is the correct. Here they are telling that finally, which one is the last step? Which one is the last step in the muscle contraction? They are telling. Okay. And binding of the calcium with the tropomyosin C and subunit of the troponin. You all know, guys, right? So this is the actin filament. On the actin filament, there is a tropomyosin is there, right? Tropomyosin is there. And on the active site of the actin filament, so there is a troponin will bind, right? Troponin. Troponin. So where the active site of the actin was, so there is hidden, right? So whenever the calcium concentration was increased, so calcium will come and it will bind to this troponin. Then troponin along with the calcium is going to be removed, right? Troponin and along with the calcium is going to be removed. So here what will happen? Our myosin head is already activated, right? It is activated by the ATP, right? So it is activated by the ATP. So now it will come and bind to this active site, right? So it will, this is the myosin head. Myosin head, it is activated already. Activated, right? So when it is activated by the ATP. So when the ATP hydrolysis was happen, on the time the myosin head is going to be activated. So then this all the process will happen. So then it will bind to this, bind to the active site of the actin filament, right? So that is the reason option 3 is the correct answer. In the option 3, what they are telling, exposing active site for the myosin on the actin, right? So this is the first step. And the second step, what will happen? The calcium will come and it will bind to the troponin, and the active site was open, right? Now the active site was open. Now the myosin head, which is activated, which will bind to the active site of the actin, right? So this is the second step and this is the third step. So that is the third option is the correct answer. The other options are wrong. Okay. So when the hydrolysis of the ATP was happen, on that time only myosin head got activated. Myosin got activated. It will happen before that only. Okay, fine. So next question, which one of the following infertility technique would be most suitable for the female with the blocked fallopian tube, but the normal ovaries and the uterus? Okay, so if you take this is the structure, right? Right? So where the fertilization of the egg will take place? So in the fallopian tube, right? In the fallopian tube, the fertilization will take place, right? So what happened here in this case, what they are telling, the female is having the, so there is a proper uterus and ovary is also good. Okay, normal uterus and the normal ovary only. But what happening here, that fallopian tube was got blocked. Okay, so then which one of the assisted technique we can do for this infertility process? Okay, so the first let us learn about the options which are given. So that is the IVF followed by the ZIP. First, let us learn about what is the IVF. Okay, 1 minute. Yeah. So what is the full form of IVF? Okay, IVF is nothing but In vitro fertilization. In vitro fertilization. In the laboratory condition, we are fusing the egg and sperm, that we will call it as the IVF, right? So in vitro fertilizing, that in the laboratory condition, right? So then coming to the ZIFT. What do you mean by ZIFT? Sorry, ZIFT, right? What do you mean by ZIFT? ZIFT is nothing but the zygote. So we are we are doing the IVF outside, right, in the laboratory conditions. So we will let it to form the zygote. That zygote will be transferred to the fallopian tube. Okay. So that we will call it as a ZIFT. Okay. ZIFT is nothing but the zygote. 1 minute. ZIFT, right? So that is nothing but the zygote intra fallopian tube transfer. Intrafallopian tube transfer. Okay. So then coming to the IUT. Okay, coming to the IUT. What do you mean by IUT? IUT is nothing but intrauterine transport. Okay, intrauterine transport. Okay. So then coming to the GIFT. What do you mean by GIFT? GIFT, gamete intrafallopian transfer. Okay. So gamete intrafallopian transfer. Okay, so gamete intrafallopian transfer. Then coming to the what is AI? AI is nothing but artificial insemination. Artificial insemination. Artificial insemination. Okay, so let us learn about this all the things. Okay, which of the technique we can do for this woman. Okay, IVF. So we are doing the in vitro fertilization in the laboratory condition. Then we are doing the ZIFT. Is that possible, guys? The fallopian tube was blocked here, right? So how can we do the ZIFT? ZIFT is nothing but, so the zygote which is formed in the laboratory condition, so that we are going to insert into this fallopian tube and the fallopian tube, right? So that is not possible because here the fallopian tube has been blocked, right? So that is why we can't do the option 1, right? 1 minute. Right? So we can't do the IVF followed by the ZIFT. So then coming to the third option, so they're telling that GIFT. Okay. So GIFT is nothing but what? Gamete intrafallopian transfer. Okay. So what happened is the gamete is not able to fertilize the egg. On that time, what they will do? So they will introduce this gamete near to this, okay, cervix. Okay. So they will introduce this gamete near the cervix. Okay. So near the cervix, they will introduce the gamete. Okay. So then that gamete, so that is a male gamete. So that male gamete will be transported to the fallopian tube. There the fertilization will happen, right? So in that case also, the fallopian tube was it is not correct, right? The fallopian tube is blocked. On that time also we can't do this, right? So that is the reason the option 3 is also wrong, right? Fine. So ZIFT we can't do because we are introducing the gamete. So this gamete only going to the fallopian tube. There the fertilization is happening. There the embryo cannot grow, right? Because it is blocked. So we can't do that also. So then coming to the AI, they are telling that artificial insemination. What do you mean by artificial insemination? So when in the normal insemination, so the sperm is not able to fertilize the egg on the time means, what we can do? We can do the artificial insemination. So there also we can inseminate artificially by introducing the sperms, right? So some any sperm we can introduce there. So there what will happen? So it is going to fertilize the egg. So that is also going to be taken place in the fallopian tube only, right? So that is the reason. So AA is also we can't do. Okay. Then coming to the second option, they are telling that, so the IVF followed by the intrauterine transport. Okay. Transfer. Okay. So what they're doing, so first we will do the in vitro fertilization in the laboratory condition. So the zygote will be formed. We will let it that zygote to develop till the blastomere stage. Okay. So till the blastomere stage. Till the blastomere stage, we will let that to grow, or either up to the above the eight cell stage. Okay, above 8 celled stage, we will let that to grow. Okay, so then what will happen? So this blastomere, we will transfer that into the uterine, right, guys? So we will transfer that into this uterine. So where the development of the embryo will take place in the uterine. Okay, so that is why. So we can do the IUT. For this IUT doing, we do not require any fallopian tube, right? So that is the reason we will do this IUT. Okay, so that is the reason option 2 is correct. Okay, we will do the IVF and followed by the IUT, transferring into the uterine only. Okay. Fine. So then coming to the 15th question, select the incorrect statement with respect to the natural killer cells. Okay. So you know that what is the natural killer cells? This chapter will come from the, this question is from the human health and the disease chapter. Okay. So option was statement 1, they are telling that option 1. So they are produced in bone marrow and are matured in lymph node to provide the acquired immunity. So let us talk with this statement first. So we are having the two types of the lymphoid organ, right? So one we will call it as the primary lymphoid organ, right? So the first one is the primary lymphoid organ, and second one is the secondary lymphoid organ, right? Secondary lymphoid organ, right? So in the primary lymphoid organ, which are the things will come? So in the primary lymphoid organ, our bone marrow and the thymus will come, right? Thymus, right? So then coming to the secondary lymphoid organ, so the lymph nodes, lymph nodes and pair patches, and the pair patches will come, right? And the this pancreas also will comes under the secondary lymphoid organ only. Pancreas, okay? Pancreas will comes under the secondary lymphoid organ. Okay, so here what they are telling, so these are produced in the bone marrow. Yes, the production will be happen, and maturation will also happen in the primary lymphoid organ only. Okay, maturation will not happen in the lymph node. Lymph node is the nothing but the secondary lymphoid organ, right? So that's why the maturation will happen in the primary lymphoid organ only, not in the secondary lymphoid organ. So that is why it is wrong. And coming to the continue of the statement, it provides the acquired immunity. No, it will not provide the acquired immunity. It will provide the innate immunity. Innate immunity. Okay. So that is the reason this option is wrong. Okay. So 1 minute. Yeah. So this option is wrong. Okay. So then coming to the, so they can act against the virus infected cell and the tumor cells. Yes. So these can act on the virus infected cell and also the tumor cell. So that is the correct statement. And option third, they are telling they produce the perforin. Yes, guys. So you know how the T natural killer cells will work. So it will produce some type of the perforins chemical. So where this perforins will create the pores in the cells. Okay. So when it is create the pores in the cell will burst out. Okay. So and it will release the all the content outside of the cells. Okay. So that is how the perforins will work. Okay. So this statement is also correct. Then coming to the options, fourth statement, they are included in the cellular barriers in the innate immunity. Yes. So these natural killer cells are under the cellular barrier in the innate immunity. Okay. You know that what is the innate immunity, right? From the birth only we have that immunity, that we will call it the innate immunity, right? Our sweat and coming to the HCl and our skin, everything will come under the innate immunity, right? Yes. So this statement is also correct. So then what they are telling here in the question, incorrect statement with respect to the natural killer cell. So the first option is wrong. So that's why it is the correct answer for this question. Okay. So the first question is the correct. Okay. So whereas others are wrong because that statements are correct. Okay. Fine. So let us go to the next question. So read the statement, a 16th question. Read the statement and select the correct option. Option A, they are telling that photorespiration does not occur in the maize plant. So you know guys, maize plant is the C4 plant, right? So the mice plant is comes under the C4 plant. Okay, it is comes under the C4 plant. In the obviously in the C4 plant there is no photorespiration, will not happen. Okay, so the photorespiration does not occur in the mice plant. That is correct statement. Then coming to the option 2, the total 18 ATPs are required for the synthesis of one glucose molecule through the Calvin cycle. Okay, yes. So see here. So for fixing one carbon dioxide, okay, for fixing one carbon dioxide, we require the 3 ATP molecule. Okay, and you know that for fixing the one glucose molecule, we require 6 carbon dioxide, right? 6 carbon dioxide require the how many ATP? So we can do the cross multiplication. 6 into 3, 18, right? 18 ATP, right? So that is the correct statement. So then coming to the option C, Rubisco is the primary carbon dioxide acceptor in the C4 plant. Okay. So no guys. So whenever you're talking about the C4 plant, the photosynthesis, how it will happen. So it is having the two types of the cells, right? So one is the mesophile cells, mesophile cell, and second one is a bundle sheet cell, right? Another one is the bundle sheath cell. Okay. So the carbon dioxide fixation will be happen in the bundle sheet cell, but the carbon dioxide which will accept, okay, which will accept will take place in the mesophile cells. Okay. In the mesophile cells, there is a enzyme. That name of the enzyme is PEPcase. PEPcase. Okay. That PEPcase will accept the carbon dioxide. Okay. That is nothing but phosphoenol pyruvate. Okay. So acceptor. Acceptor. Okay. So why we will call it as a C4? We all know that. So because the first product which is formed in the C4 plant is the, so that is having the 4 carbon. Okay. So that is why we will call it as a C4 plant, right? So the first acceptor is the PEPcase. So that is nothing but the phosphoenol pyruvate. So that enzyme is PEPcase. That substrate is the PEP. PEP. PEP is nothing but phosphoenol pyruvate. Okay. Phosphoenol pyruvate will accept the carbon dioxide. So then it will convert into the C4. Okay. Fine. So that is the reason. So the correct, so this Rubisco is primary carbon dioxide acceptor in the C4 plant. No. So that is not the correct statement. Okay. So then coming to this thing, they are telling that in the option 1, all A, B, C are correct. No, C option is wrong, right? So that is the thing. Option 1 is wrong. Then coming to the option 2, only A and B is correct. Yes, option A and B is correct, right? Yes, option A and B is the correct. So then coming to the third option, they are telling that only C is correct. No, only C is only wrong, right? So that is wrong. So then coming to the option 4, they are telling only B and C is correct. No, C is wrong, right? So the correct statement is only A and B is correct. Sorry, some disturbance. Okay. So that is why the correct option is A and B only, right? Okay. So, you know, guys, in the bundle sheet, what will happen? Carbon dioxide fixation will happen. Carbon dioxide. Carbon dioxide fixation will happen. Okay. So that is in the bundle sheet cell. Okay. So in the bundle sheets only, the Rubisco is also going to be present. Rubisco is also going to be present in a bundle sheet cells only. So the correct answer is option 2. Okay, fine. So let us go to the 17th question. So the nitrogenase enzyme. So this is from the chapters. So that is the minerals. Okay. So minerals chapter, mineral nutrition chapter, this question. Nitrogenase enzyme as Fa and MO element. And in the statement 2, they are telling that yes, the nitrogenase is having the Fe and MO element. That is the correct statement. And option 2, they are telling that it resembles for conversion of, it is responsible for conversion of NH3 to the 2NO, sorry, 3NO. No, it is responsible for conversion of N2. So that is a dinitrogen, right? From the dinitrogen N2 to the NH3. right? So it will give N2 to the 2NH3. Okay. So that is the reason. No, it is not related to the conversion of NH3 to the 2NO. So that statement is wrong. So then coming to the option 3, it is present in eukaryotes only. No, it is present only prokaryotes. It is present in prokaryotes. Okay. It is present in prokaryotes. So that is the statement is wrong. Then coming to the option 4, it has a copper as a cofactor. No, it has the Fe as a cofactor. So that is the reason. That is wrong. So the correct statement, the nitrogenase enzyme is having the Fe and MO as the MO as the MO as the cofactor, and it will convert dinitrogen into the two molecules of the ammonia. Okay. So that is the reason option 1 is correct, option 2 and option 3 and option 4 is wrong. Okay. Fine, guys. So let us go to the next question. So they read the following statement and choose the correct answer. Option A, they are telling that here the two statements are given. So you have to select which one is the correct. Okay. Heliocecal valve present, it will prevent the backflow of the fecal matter from the large intestine into the helium. What they are telling, so here you can see the structure. So that is a small intestine is made up of the three main structure, right? Helium. So here helium is the large structure, right? Helium. Here you can see helium. So helium is connected to the large intestine. That structure we can call it as a cecum. Okay. So here you can see the helium. So here this is connected to the, here it is connected to the cecum. Okay. This is the cecum structure, right? This is cecum. So it is the helium. Okay. So here there is a structure is there, right? There is a valve is there. This valve we will call it as what? Heliocecal valve. Okay. So that's that valve we will call it as heliocecum. Heliocecal valve. Heliocecal valve. Okay. So what is the main function of this? One, the food, which is the undigested food that is enter into the large intestine. So where this heliocecal valve-like structure, it will be valve-like structure. So it will close. Okay. It will close. It will not allow the backflow of this undigested food into the small intestine. Okay. That is correct. That is correct. Okay. Then coming to the option B, in a statement B, they are telling marasmus generally found in the infant. So, you know, guys, there are the two types of the protein deficiency are there. One is the marasmus and another one is the kwashiorkor, right? Kwashiorkor will be for the above the 1 year of the infant. So that is when, so we are not given the, so when the mother milk was replaced by the high calorific value food, but not the proteinaceous food. Okay. So here you can see marasmus generally found in the infants below 1 year of age. That is correct. If mother milk, when it is going to be form the marasmus, when the mother milk is replaced too early by the other food. Okay, that other foods which are low in protein and both calorie. Okay, when the food was both low in calorie and protein, so you will get the marasmus. The infant will get the marasmus. So that is before, so that is below 1 year age. Okay, so and coming to the kwashiorkor, that is for above 1 year age. Above 1 year age, okay, and it is replaced where high caloric food is there. High caloric food is there, but no protein. No protein. Okay. Yes, the statement 2 is correct, right? So that is why let us see that. So both the statements are correct. Both the statements are incorrect. That is not. And only A statement is correct. No. Only B statement is correct. No. So both statement A and B are correct, right? Yes. So that is why option A is correct, whereas the option 2, option 3, and option 4 are wrong. Okay. Fine. So let us go to the 19th question. Choose the incorrect statement with respect to the secondary immune response. Okay. So let us go to the option 1. So the vigorous immune response against a pathogen on exposure to the same antigen repeatedly, right? So whenever we are talking about the secondary immune response, when we are first time when it was when the virus or any bacteria it is visited our body. So when it is infected first time, the response will be slow, right? So our immune system will be very slow. On the time we'll get the fever for 1 week, then will we get recovery, right? So do you observe whenever we are so when the first time the coronavirus will infected us on that time, so it is take the so many time, right? It will take like the 1 week to recover, and for the 10 days 12 days to recover, right? So but whenever the second time it is exposed to our body, so on that time what happened, we recover very fastly. Within 2 to 3 days we will recover, right? So that is the reason. So here you can see within like 4 to 5 days of the time we will recover, so in the second exposure, right? So here they are telling that vigorous immune response against the pathogen that is exposed same antigen, that is the same antigen repeatedly. Alva. So that is the reason the option 1 is correct. Okay. That statement is correct. And coming to the option 2, they are telling that the secondary immune response will be slow and it is short-lived. And again the pathogen which is entering again and again repeatedly in the body. So first statement only telling that, so whenever the antigen was repeatedly it is infecting, so the response will be fast, right? In the option 2, they are telling that opposite of that, right? So the first statement is correct, whereas the second statement is wrong. It is not slow, it is a fast, right? And it is not short-lived, it is a long-lived, right? Because the memory is going to be formed, right? It is a long immune response is triggered by the memory cells is formed. So that will be helping. So that is the, it is also called as quick, okay, quick response. Quick response. Yes. So that anamnestic response is very quick. So that is the correct answer. Okay, that is the correct answer. So in the question they are asking that incorrect statement, right? So 1 and 3, 4 are the correct statement. Which one is the wrong? So the wrong statement is the second one, right? So the second one is the wrong. So that's why second one is the correct answer. Okay, the first one, third one, and fourth one is the wrong. Okay, guys? Fine. So let us go to the 20th question. Identify the incorrect statement. You have to identify incorrect statement here. Means they are given the three correct statement. Three correct statement is there. So one wrong is there. In the wrong statement, you have to identify. Okay. So the gene for beta carotene was extracted from the daffodil. Okay. So daffodil is nothing but a plant. So that will be yellow in color. Yellow or some will be orange in color. Okay, so this yellow in color, yes. So the beta carotene can be extracted from this beta carotene. Okay, so yes, the beta carotene can be extracted from this. So that is the correct reason. So that is the correct question. Sorry, correct statement. So then coming to the option 2, GM plant Brassica napios produce a hirudine. Yes. So the that is the genetically modified plant. So that is the Brassica that will produce the hirudine. So that is acting as the anticoagulant. Okay. It is acting as anticoagulant. Okay. So do you know whenever the leeches will bite us, the blood will be flow like flow, right? So on that time we will use this anticoagulant hirudine. Okay. So we will use this hirudine on the time the blood will not flow. So where the coagulation will happen. Okay. So that's why we will use this hirudine. So that is why it is a correct statement. Okay. So then coming to the option third, so that is the third statement. Bt toxin enters the ovary of insect and causes sterility. No. So what is the function of Bt toxin? In the biotechnology, we have learned that. So the Bt toxin will not enter the ovary. So Bt toxin will enter the midgut, right? Midgut of the insect or the pest. So where in the midgut, it will destroy it will cause the pores, right? It will cause the pores in epithelial cell. Pores in epithelial cell, and it is causes the osmotic lysis, right? It is causes the osmotic lysis, thereby it is causing the death of the insect, right? Death of the insect. It is not causes the sterility. So this is a wrong statement, right? So then coming to the PCR, so that is nothing but the polymerase chain reaction, right? Polymerase chain reaction 1 minute. Yeah. Polymerase chain reaction. So that is a molecular diagnostic tool. So we will use for the diagnosis of the diseases. For example, if you take the RT-PCR, right? So restriction PCR. So that we will use it for the diagnosing the coronavirus, right? So many PCR types we will use for the diagnosis of the some of the diseases, and AIDS. AIDS also can be recognized by the PCR, right? Yes. So that is why option third is also correct. Okay. So here in the question they're asked that incorrect statement, right? So that is the reason option third is the wrong. Okay, that is the wrong statement. So that is why option 3 is the correct. Okay. Okay, guys. Fine. So let us go to the next question. Read the following statement and choose the correct. Okay. Correct answer. So let us try. So in the MOET, so what is MOET? Multiple ovary embryo transfer. Okay. So what do you mean by MOET is MOET is nothing but multiple embryo. Sorry, multiple ovary embryo transfer. Multiple ovary embryo transfer. Okay. So they are telling that in the multiple ovary embryo transfer, a cow is administered hormone with the estrogen-like activity. No, it is ingested with the administered with the FSH-like activity, right? FSH. FSH is nothing but the follicle stimulating hormone, right? So it is injected with the FSH-like hormone, not the estrogen-like hormone. Okay. So to produce the follicular maturation and the super ovulation. What will happen in the MOET is, so here what we will do for the cow, we will inject the, so artificially we will inject the FSH. Okay, so hormone FSH. What will happen now? So many ovary gets and so matured. Okay, so what will happen here? So many ovary get matured here. Okay, so that is why we are calling as multiple ovary. So once a time means in the when the menstruation is happening. So on the time, what will happen? So FSH will give rise to the only one egg, right? So, but what happened in the Easter cycle here? So, once the FSH was administered more, if it is administered more means, so super ovulation will happen. Okay, so many ovary will be developed here. So that is what we will call it as a super ovulation. Okay, so follicular maturation and the super ovulation will happen. So that is a wrong statement, right? Because we are not using the estrogen, we are using the FSH. Then coming to the say statement number 2, the success rate of crossing mature male and the female animals is fairly low, even though the artificial insemination is carried out. That is right, right? So whenever the age was more, so whenever the matured male and the female animals is crossing on that time, so if we do the artificial insemination also, the that sperm movement will be very low. Okay, the sperm movement will be very low on that time. So that is the reason option B statement is also wrong. Okay, so sorry, the B statement is correct. 1 minute. Sorry, guys. Yes. So this the statement is correct. So the success rate of the crossing mature male and the female animal is a fairly low in the case of artificial insemination carried out also. That is correct. Right. So that is the reason only the option B is the correct. Okay. In the one statement they are telling both statement are correct. No. In the option they are telling both statement are correct. No. Only A statement is correct. No. And only B statement is correct. Yes. Only B statement is correct, right? Yes, guys. So other than the B statement, so other options are wrong. Okay. So that's it, guys. So if you have any questions, you can ask us in the group. Okay. So thank you so much. Let us end here. Thank you, guys.